Hello everyone, my name is Poonam Vinayamohan and I'm a PhD student at the University of Connecticut. So today I'll be presenting my research which is titled Effect of Plant-Derived Antimicrobials in Disrupting Horizontal Transfer of Antibiotic Resistance Gene between Multidrug Resistant Salmonella and commensal E. coli in broiler chicks, both ex vivo and in vivo. There are trillions of tiny bacteria all around us. Even though a majority of them are harmless bacteria, there are quite a few which can cause severe diseases in humans, such as salmonellosis. Fortunately, we are able to treat these bacterial infections using antibiotics. However, like any other organisms, the bacteria has also developed certain mutations, some of which can provide resistance against antibiotics. So in the presence of antibiotics, the non-resistant bacteria get killed off, providing extra room and resources for the drug-resistant bacteria to thrive, passing along only the mutated resistant genes, which help them to do so. Now, reproduction is not the only means by which they can do this. There are certain bacteria which can donate the DNA once they die off, while there are certain other bacteria which can exchange their genes through a process called as horizontal gene transfer or conjugation. Over time, these drug-resistant bacteria, they proliferate, creating an entire new strain of drug-resistant superbugs. Now, how long do we have before these superbugs take over? Well, sadly, it has already occurred in several bacteria, which include Salmonella. Now, Salmonella heidelberg is a very common strain of Salmonella which causes foodborne infections in humans. And most of the Salmonella heidelberg infections, they are related to poultry-derived food. And the peculiar thing about Salmonella is that they are resistant to more than one type of antibiotics. Now let's see how the horizontal gene transfer or conjugation happens in detail. Now, as a donor bacteria, we have a multi-drug resistant pathogenic bacteria, which is Salmonella. And here is the blatam gene, which provides resistance against antibiotics. And as a recipient, we have an E. coli, which is a harmless, non-resistant bacteria. So once conjugation happens, a conjugative bridge is first of all between the two bacteria. Now this conjugative bridge is actually formed by the fusion of bacterial pilus, which are actually hair-like appendages that are seen on the surface of bacteria. Now once the conjugative bridge is formed, a copy of this blatam gene gets transferred into the recipient bacteria. Now this E. coli has this blatam gene and thereby it has become drug resistant. So how can we prevent this from occurring? So our in vitro studies have already proved that we can use several plant-derived antimicrobials, which specifically target the genes that are involved in conjugation. Now, what are these plant-derived antimicrobials? So these are natural substances that are produced by plants as a defense mechanism against plant pathogens. So in this study, our primary objective is to see whether we can use these PDAs for reducing the gene flow that is occurring in the poultry gut. Why poultry gut? So poultry gut is considered as a reservoir for many drug-resistant pathogens as well as genes. And these bacteria, they are constantly interacting with each other as well as they are exchanging the genes between them, creating an entire gene pool of antibiotic-resistant genes. Now, how can we prevent this from occurring using the PDAs? It's very simple. We can feed the chicks with the plant-derived antimicrobials that are mixed in their feed and see whether we can reduce the gene flow by enumerating the number of drug-resistant bacteria before and after the treatment. And from our results, we are able to prove this hypothesis. So in the ex vivo study, we are able to see more than 50 to 90% reduction in the gene flow between Salmonella and E. coli when compared to the control. And in the in vivo study also, we found more than 20% reduction in the gene flow between these two bacteria. Therefore, our results suggest the potential use of PDAs for reducing the gene flow, as well as reducing the number of drug-resistant bacteria in the poultry gut, thereby improving food safety. With this, I would like to conclude my presentation, and I would like to thank you all for attending this presentation.